So now I would like to move on to proteomics and introduce our next speaker, Dr. Alexander Martin, University of Vigo. So Alexander, if you are ready, you can share your screen.
Hi. Hello. Can you hear me?
Yep.
Cool. I'm gonna try to share.
Now we can see your screen.
Okay. And is it full screen? No? Yeah. Nice. Nice. All right. Well, good afternoon to all. Well, I am here to talk about. Well, this talk is based in the theory that is behind all my practical experience on the field of proteomics. And my intention is not to cover all the theory behind and all the possibilities, but highlight the useful applications that I found using proteomics. That can go from discovering biomarkers of exposure of pollutants, or to try to um, find some complex modes of action. Well,
Alexander, I think we cannot hear you.
Really useful because gives us more specific information. But we are keep having the general information that can be deduced from the genome relationship. So why proteomics? Well, in detail, of course, genome does not describe the dynamic processes that the proteome can reflect, and also does not sustain biological functions. Studying transcriptome, and when it's compared with the proteome, shows really low levels, well, not really, but low levels of correlation, around 40% of this new information that is not reflected. So both levels could be really useful. And also we can still trace the origin and deduce the functions of the proteins in a more general way. In the case of proteins, every time that we jump one step of these levels of organizations, we are getting more variability. Here I bring a relatively simple case that is a virus that only with 12 genes can produce more than 40 proteins. And of course, if we start to consider the modifications that the proteins can have, the number increases. Orders of magnitude. When we see the numbers for human genome, the numbers respond in the same way. Most of the steps jumps like 1 order of magnitude. So, well, I think we all know, but just to define the term of proteome, exist two different definitions. The first one, more theoretic, is based on, it's inspired on the genome word, and would be the whole potential proteins that we can find in a specific organism. This would be all the proteins in the different stages, different ages, different organs, and all the proteins that can appear here. But much more common working definition of proteome is just the situation, the photography of the situation of the sample when it's taken. I like to call it long exposure photo compared with the usual word for transcriptomics that is a snapshot. And I like to do this difference because proteins move slower, change slower than transcripts. The previous speaker says that only in 5 days they can find beautiful changes in the transcriptome. Proteins are still longer. And well, we are working with 7 days and we still found difference in the proteome. But of course, it's important to consider that the changes will need longer time, but also will stay for longer. So the photo that we are taking in this case is like a long exposure photo. And of course, for the application of proteomics in ecotoxicology, the important part or the more useful tool is the differential proteomics, when we can compare different treatments. So proteomics in the context of ecotoxicology has also different levels of complexity that will require different skills and also will answer different questions. In the first case, we can be just in the detection and quantification of proteins, but we can jump and we can go for post-translational modifications of these proteins that can be detected also. And in the last stages, we can go for interactions of proteins. Of course, the number of studies are much more common for protein detection based on protein detection and quantification and decrease when you go further. So when we are jumping through these different levels, you can answer more specific questions. So here I'm gonna go into the details of massive protein detections. Proteomics can have target detections, but here, like this workshop is for omics and massive. I think that is the most interesting part. And also it's based in my experience and in my methodological approaches. But to achieve an experiment based on massive protein detection, you need to separate and extract the proteins. In this case, it's pretty similar to when you are extracting RNA, but we are keeping the lower part is a way to quantify the proteins that we extract. In the second stage, we need a method for separation. It can be done in a gel. In this case, to the 2-dimension gels to achieve a separation of the proteins, or it can be done through liquid chromatography. In this case, liquid chromatography can be directly attached to the detector, in this case a mass spectrometer, and can be done directly. And the last step, of course, is the detection in the mass spectrometer. Going into the details of the detection, in this case, I think it's a different when we are studying metabolomics, that as far as I know, we stay just in a first detection in the mass spectrometer. With proteins, we can go further and we can go in a 2 rounds mass spectrometry. It is called MS, and we can go further into the details of what we are putting in. So this is a spectrum from the results of one particular peptide. And it can be deduced, it can be identified which peptide is which. There are 2 methods, the de novo method that can deduce the particular amino acids based on the proportion of the mass and the charge that we are in the different peaks. And this is independent of the database. So it's a really, really interesting approach. But the data here is is split with the charge. And sometimes the particles that we are introducing can have some variability on the charge. Most of the time, it can be deduced because we know the mass of the different amino acids, and we can reconstruct, but different combinations of the mass, the M will represent the mass of the amino acid. Sometimes it can be double or even more, and the charge can be also double, triple. So this theoretical mass and charge will change. And in some cases, it cannot be resolved, the sequence of the particular sequence of the amino acids. To resolve that and improve the number of identification, we can go through the fingerprinting. And we can recreate in silicon the theoretical spectrum that a change of amino acids will bring. And in this case, comparing with the real spectrum, we can know if it's belong to several peptides. In this now we are in the identification, but also we can quantify the number of peptides and the number of proteins that we have in the sample. To do that is a really useful and really usual approach to use multiplexing. Multiplexing is a strategy that involves adding some labels. In this case, labels that has the same mass to the different samples. So each sample will be labeled with different molecules. These molecules at the beginning, for the first detection in the mass spectrometer, will keep the same mass. So it's going to be no difference at all in between samples. And after the first detection, it's going to be a selection of individual peaks. Each of these peaks of this spectrum will be keep going in the spectrometer. So when the spectrometer select one of these peaks, we go to the next MS in this case, and we are getting the particular spectrum of each fraction. In this part, the label will break in a particular way, and each label will break in a different mass pieces. So when we study the spectrum of the second MSMS, we can explore the low mass particles. That here we have a zoom of this part of the graph, and we can found the labels break in different way, and the intensity of the labels will give us a relative quantification of the samples in the same run, in the same time. So, well, we need to go then through a normalization process. Most of the time, the normalization is done by total amount of protein. And, well, each of these columns will represent one label and one sample, and the whole bunch of proteins that we can detect. In this case, in the first approach, we can go for the total protein and make the normalization. But if we want to compare in between different experiments, different runs of the mass spectrometer, we need to add another channel, another label in this case, that will be a control. Usually is a mix of the rest of the samples from different experiments to match this column in the two, in this case, in the two experiments. So this will be an inter-normalization plus the intra-normalization that it needs to be done. And then in particular cases, just to show you and not go deeper in this, but in particular cases, it's important to do another normalization, especially if the abundance of the proteins jump a lot of orders of magnitude. But well, this is just the common ways that we can normalize all the proteomic data. once we have all these data available, we can go through the enrichment analysis in the same way of transcriptomics. We go through a functional annotation with a bunch of different Go terms, and then through a specialized software that is based in Fisher tests, we can compare if the proportions of the different groups are the same or not. And in this case, we can extract the different Go terms that change in the treatment. So now, with all of this, I want to show you what we can extract from all this data. This is a little spam about my biological model. This is the sheep's head minnow that we are using it in Europe. This is typical from US. It was a really, it is a really good standard for ecotoxicology. And here we are trying to bring some representativity to use it in Europe. And these are particular examples of different experiments. I don't pretend to explain the meaning of this, but just to show you how the data can show us biomarkers or other kind of results. So in this case, it's quite clear, well, this is a volcano plot, the same kind of representations that we can find in transcriptomics. And it's quite clear that this cloud of dots, that each dot is a product of, in this case, estrogenicity, but well, biomarkers of exposure of this pollutant in particular. Well, it's quite clear, no? It's separate from the rest of the data. And now we can go further and go into the pathways, but I wanna show a warning because well, this is a term that it has been used in previous talk, that probably from here is gonna be interpretation fatigue, no? But I'm not trying to overwhelm with the details, just to show you the potential of this technique. But if we jump directly through the pathways, we can find that most of the proteins that were in that cloud that I showed you in the upper part of the volcano plot are just here. And are directly related with the activation, in this case, of one mechanism, one receptor in particular. But the important concept from here is that we can deduce alterations in particular pathways. If we generalize this idea, we can go through all the pathways that can be affected in our treatment. And each of these ones, we can click here and go into details of more and more pathways. That will bring us a really good picture of what is happening. So, summarizing all of this, we can work with the most affected proteins, like biomarkers, or try to validate if these biomarkers can be used for specific pollutants. Also, using all the information of affected pathways and the information from the enrichment analysis. And also, and this is really important, working with the background knowledge, we can try to deduce different modes of action of unknown pollutants, for example. And this is a specific example from one of my works that, for example, using choronin 1A is a specific protein that maybe is a candidate of exposure or a candidate of biomarkers of exposure of particular molecule. Also studying the different Go terms affected around this protein and this exposure, and also checking that there are processes related with the stability of the membranes of different kind of cells or mitochondria. We can infer that the mode of action of this pollutant is the alterations of the stability of the membranes. So this would be an example of that can summarize how to extract all the information, the different levels of proteomics. And well, to-dos for proteomics in ecotoxicology, the same as the question before, no? What happened with non-model organisms? Well, depend, of course, in the knowledge of the non-model organism, but there is, it is important that the annotations has sense. Otherwise, you can find, for example, alterations in neurons when you are using a liver, for example. It can be maybe because of the nerves that are present in the liver, but much more probably is because the same proteins that are related in neurons are related also in the cells of the liver, and it contains the same GO terms associated. So when we are performing enrichment analysis, there are some GO terms that are not related truly with the nature of the samples. Also, it's important to improve the function, the knowledge of the function of the different proteins. For example, we found an interesting candidate for biomarker of exposure of estrogenic compounds in fish, that the main function of this protein is to be in the milk of mammals. So of course, in this case, for marine fish, has no sense at all, and the function is still unknown. No? And then I think a really interesting potential for proteomics is the study of post-translational modifications. The most easy application for that, I think, is to try to identify if specific biomarkers has specific modifications, and this will bring us much more than we have found biomarkers much more specific, but also the study of general alterations in the post-transcriptional modification would be really interesting to make it in a standardized way. Then thing that I saw that is a lot of people working is how to interactions of proteins through different pathways, and also the standardization of this analysis. No, I will say also that reproducibility is a really important thing to do in proteomics for ecotoxicology, and improve the meta-analysis also. As I saw that previous talks were talking about some kind of algorithms that can integrate different levels for the same results. So I would say that it's important also, it's important to do to integrate proteomics in this kind of meta-analysis. And with that, I want to say thank you. And if you have any question, please.
Thank you so much, Alexander. It was a really interesting talk, and we are really happy about having early careers in our speaker list, as well as in the committee. So thank you for that.